Welcome, everyone, back to the wastewater stream, EOCP 2021 Day 2. This next session is from 3 to 3:45, and we're going to discuss microbiological monitoring of BioP systems. And we have Tony Glimpf-Martin presenting for us. She's presenting virtually. I'd like to tell you a little bit about Tony. Tony's an environmental microbiologist specializing in water and wastewater treatment and treatment process consultant with wastewater microbiology solutions. She provides technical assistance, consulting services, and microscopic evaluations for more than 30 industries and 80 municipalities. Prior to that, Tony served as a senior level supervisor in the monitoring and research division of the Metropolitan Water Reclamation District of Greater Chicago, and has more than 40 years of water quality related experience. Tony has conducted over 400 technical workshops and has made numerous presentations at conferences, public meetings, the Great Lakes Commission meeting, and for state representatives. We're so happy to have Tony here, and we look forward to her presentation. Thanks, Tony.
Thank you. I have to say something really quickly. I've been in this for 40 years, and I was 2 years old when I got started. Okay, so let's talk about microbiological monitoring of biological phosphorus removal systems. There are basically two ways that phosphorus get removed from wastewater treatment. One is there's two sources or two types of phosphorus. There's particulate phosphorus, and then there's the soluble phosphorus. The particulate phosphorus can be removed through solids removal. The soluble phosphorus can be removed two ways: one by precipitation, chemical uptake, or by microbial uptake. So we're going to talk about the microbial uptake. So biological phosphorus removal is accomplished by phosphorus accumulating microorganisms. You're going to hear a lot of acronyms. And these are PAOs, phosphorus accumulating organisms. And they accumulate phosphorus in the form of polyphosphate. These are microorganisms that naturally occur in the environment, and they naturally occur in our treatment process. So they're always there. But our job, when we want biological phosphorus removal, is to provide an environment that's going to cause them to dominate in the system. So PAOs are their group of bacteria that, under certain conditions, they facilitate the removal of large amounts of phosphorus from the wastewater. They can accomplish this by accumulating it in their cells, and they do it in the form of polyphosphate granules. But they're not the only bacteria that can accumulate polyphosphate within their cells. This is a widespread ability among bacteria. There's a whole lot of different types of bacteria that can do that. There are filamentous bacteria that have the ability to store phosphorus. But PAOs have a characteristics that other bacteria do not have that make them a little bit more suitable for this biological phosphorus removal processes. They can consume simple carbons, and when we say carbons, we're talking about organic material. So they can consume simple organic material in the absence of nitrate, in the absence of oxygen, in other words, under anaerobic conditions. And this gives them a preferential access to the simple compounds that come into the wastewater treatment plant. So how many of us have seen this diagram? This is biological phosphorus removal. I don't have a clue what this means. So I'm just going to make it very, very simple for you. So in the biological phosphorus removal process, the growth of PAOs is encouraged by cycling them from anaerobic conditions to aerobic conditions. So we got to take them from anaerobic to aerobic and back. And so what happens in the anaerobic zone is you've got complex organic compounds are broken down into simpler forms. And then those simpler forms, they take those simple compounds and they produce the volatile fatty acids. So in other words, just kind of imagine mama bugs that crack open the pecans in the shell so that the baby bugs can eat the pecans. And then once they eat the pecans, they produce the volatile fatty acids. So you got the degraders that break up the complex compounds. And then you have another set of bacteria that take those simplified compounds and they produce volatile fatty acids. And so those volatile fatty acids are immediately used by the PAOs. If you want biological phosphorus removal, you have to have volatile fatty acids, because the PAOs take these volatile fatty acids and they convert it into their bodies in the form of PHB. Okay, I'm going to make it a little bit more simple for you. So imagine this is a PAO. PAOs already have phosphorus in their body. Most bacteria already have phosphorus within their cells. And so there's VFAs in the anaerobic zone. This is the anaerobic zone. So a phosphorus bug, a PAO, if he sees the VFAs, he thinks to himself, well, I want to eat it. But he doesn't have the energy to eat it. So what he does is he takes that phosphorus and he busts up that phosphorus, and that gives him the energy to uptake that VFAs. But when they store the VFAs in their bodies, they store it in the form of PHB. So what he did, in essence, was he took the phosphorus that he had in his body and he exchanged it for PHB. This occurs in the anaerobic zone, but it only occurs if there's VFAs, because he takes those VFAs and he stores it in his body in the form of PHB. So now when you take him into the aerobic zone, he's got all his PHB in his body. So when he sees phosphorus, he thinks, oh, wow, I really want to eat it, but I don't have the energy. So what he does is he busts up that PHB, and then he can uptake an enormous amount of phosphorus. So now we cycle back to the anaerobic zone. Now he's got all this phosphorus in his body. He sees VFAs, he wants to eat it. He said, I don't have any energy. So he busts up that phosphorus, and then he can uptake those VFAs and store it as PHB. That's why in the anaerobic zone, you'll see this release of phosphorus. And so what you need to understand is that phosphorus uptake in your aerobic zone is directly related to the amount of PHB that's stored in that anaerobic zone. So if these microbes don't have PHB in their bodies, when they get into the aerobic zone, they're not going to take up phosphorus. So these two are related to one another. And so when you want to look at these microscopically, they can be visualized by just conventional microscopy. You don't need any electron microscopes or any fancy microscopes. In the anaerobic zone, PHB is a lipid-like polymer. And so what the Sudan black stain does is it stains that lipid. It stains the PHB. And so you can see the PHB granules in the anaerobic zone just by using the Sudan black stain. And so it'll look something like this. Most PAOs like to grow in clusters. And so this is in the anaerobic zone. Why do I bother to look at these in the anaerobic zone? Because if I don't see PHB in my anaerobic zone, I'm not going to get phosphorus uptake in my aerobic zone. And then I'll tell you another reason why shortly. So here's another just image of just a large cluster of PAOs. This is the PHB. This is in the anaerobic zone. So in the aerobic zone, where phosphorus uptake occurs, the phosphorus is stored in the cell as polyphosphate granules. And so you can't see these granules unless you use a stain. And so the stain method that I use for the aerobic zone is the Neisser stain. Because what the Neisser stain does, it stains polyphosphate. So this is a cluster of PAOs that you can see in the aerobic zone. Here's another example. It kind of looks just like a big cluster of grapes. But there's another bug that kind of gets in the way, and this one we call GAO. These are glycogen accumulating microorganisms. So in the anaerobic zone, there's this undesirable group of microorganisms. They compete with the PAOs for the VFAs. They like the VFAs just like the PAOs like the VFAs. But they look a lot different. You can notice they look like fish eggs. They are hollow, very hollow. Most GAOs are large cells. They have the ability to take up VFAs under anaerobic conditions, but they are unable to accumulate phosphorus under aerobic conditions. So just like PAOs, GAOs are capable of taking up the VFAs, but they don't contribute to phosphorus removal. So if I see a whole lot of GAOs, what they're doing is they're taking up all the food from the PAOs, and they're not doing any work. So if you look at this image, at the top are PAOs and at the bottom the GAOs. And in the anaerobic zone, the PAOs, the GAOs, both of them, they see the VFAs, they want to eat it, they don't have the energy. Your PAOs will break up the phosphorus, they'll have energy, they'll store it as PHB. Your GAOs, they break up the glycogen for energy, and then they take up the VFAs and store it as PHB. So if I'm looking at my PHB stain in the anaerobic zone, I don't know if it's a GAO or if it's a PAO, because what I'm staining is PHB.
Hi, Tony. I'm so sorry to interrupt you. I just didn't know if you wanted to be sharing some slides with the audience. We aren't able to see any PowerPoint slides. I didn't know.
Oh, you were really? I'm looking at them. I thought they were. I'm clicking through.
Yeah,
I'm sorry.
No worries. You might have to click share. Sorry, we.
Oh, okay.
Share screen.
Sorry. I'm sorry. You mean I did all that talking and nobody could see me? Okay, I'm gonna go back a little bit because I want them to see these images.
Oh, we'd love that. Thank you so much. You know you're pressed for time, but
let me share I'll just talk fast. I'm so sorry. Let me see here.
Sorry, I didn't interrupt earlier.
Oh, okay. Okay, so I how do I get back to share? I was at share. I thought. Okay, here. Okay.
Yeah.
Can you see it now?
There you go. Yep.
Oh, I apologize.
No worries. Oh, perfect. And in presentation view. Oh, beautiful. Okay. All back to you, Tony. Thank you.
Okay. Okay, so this is what I was talking about in the anaerobic zone. With your normal PAOs, they're in the anaerobic zone. They have phosphorus in their cell. So they're in the anaerobic zone. They see VFAs. They don't have any energy. So they bust up the phosphorus, and then it can take up the VFAs, and they store it in their body as PHB. So once they go into the aerobic zone, now they have a lot of PHB in their bodies. And so then when they go there, they want to take up the phosphorus, but they don't have energy, so they bust up the PHB, and then they can take up phosphorus. And then when they recycle back, now they've got a whole bunch of phosphorus. They take up the VFAs, and they store it as PHB. So, like I said, the phosphorus uptake in this anaerobic zone is directly related to how the phosphorus uptake that you get in the aerobic zone is directly related to how much PHB you have in the anaerobic zone. Okay, so like I was saying, you can see the PHB microscopically using the PHB stain, and so it'll look like this. You'll see this cluster of cells. PAOs like to grow in clusters. So here's another image of the PHB in the PAOs. And this is what we take in the anaerobic zone. In the aerobic zone, we use the Niger stain. And so it looks more like this in the aerobic zone. Because what the Niger stain does is it stains the polyphosphate granules. The PHB stain stains the PHB in the anaerobic zone. The Niger stain stains the phosphate in the aerobic zone. And so you can see this cluster. Okay, then we were talking about this other undesirable group of microorganisms called GAOs. And if you look at them, they look very hollow. It's because they don't take up phosphorus. They take up all the goodies. They take all the VFAs. But when you put them in the aerobic zone, they don't take up phosphorus. So this is a Niger stain that stains polyphosphate. If you look, their cells are hollow because they're not taking up phosphorus. And this is another image of PAOs. They look like fish eggs. So they're capable of taking up the VFAs, but they don't do the work. So they serve to remove VFAs, but they don't do the work of taking up phosphorus. And so if you look at this image, at the top are your PAOs. They have polyphosphate. At the bottom are the GAOs. They have glycogen. So you put them both in the anaerobic zone. Both of them want the acetate. They both of them want the VFAs. And so they need energy to do that. Your PAOs bust up phosphorus. They take it up, store it as PHB. The GAOs, they bust up the glycogen and then they take it up as PHB. So I have no way of knowing which one it's in here. If I do my stain, if I take a sample from the anaerobic zone, I'm just going to see PHB. So I don't know if it's GAOs or if it's PAOs. So then when you bring them to the aerobic zone, your they both have PHB in their bodies, but your PAOs, they're going to bust up the PHB and they're going to take up phosphorus. But your glycogen bugs, they're going to bust up the PHB and they're going to take up glycogen. And so when you bring them back around, the same thing happens. PAOs take up PHB, GAOs take up PHB. So here's a better way to look at that. So my family are the PAOs. Okay, so I have 10 hamburgers, and I have to go to eat 10 hamburgers in order for me to go to work to make $10. So then I take those $10, and then I can come back and I can make 10 hamburgers. But now my in-laws came to live with me. So they take 3 of my hamburgers. So now I only have 7. So when I go to work, I only can make $7. My in-laws are the GAOs, by the way. And so they take the $3 hamburgers, and they don't go to work. They just sit on the sofa. Okay. So now when I bring them back around, they took 3 more of my hamburgers, and now I only have 4. So when I go to work, I'm only going to make $4. But they're going to take and eat 3 of my hamburgers and sit on the sofa and do nothing. Then we bring them back around. They took 3 more of my hamburgers, and I only have 1. So when I go to work, I only can make $1. They sit on the sofa and do nothing. So that's kind of what happens when the GAOs come. They take all the food, but they don't do any work to help remove any phosphorus. And so if you ever look in a sample and you see this many PAOs and that many GAOs, then you can just about guarantee you're not getting good phosphorus removal. Okay, so another thing that gives GAOs an advantage is that they can take up sugars. They can take up simple sugars. They don't have to have acetate to store PHB. They can take up sugars and they'll store PHB. So they can grow 2 ways. They can grow with the VFAs and they can grow with simple sugars. And so they can keep on multiplying and then they can eventually dominate in the system. So when I do a microscopic assessment, what I do is I collect a sample from the anaerobic zone and I collect a sample from the aerobic zone. And then I split the sample because I want to know the concentration of my sample. I want to know the concentration of the sample that I'm actually looking at. And so then I take my slide and I mark my slide about 2 centimeters apart. Why 2 centimeters? I just pick 2 centimeters. But just whatever you do, just keep it consistent. So what I do is I use a wax pencil to mark my slides. And the reason why I use wax pencil is because it's kind of raised off the slide. Because if you think about it, we're taking a few drops out of billions of drops. And so we want to be as consistent as we can. So I always use 1/5 of a milliliter, 4 drops, the same thing. 4 drops is 1/5 of a milliliter. Whatever you use, always use the same volume all the time. But what this does, having this wax lines, it contains the sample within a fixed area all the time. So you're always scanning the same fixed area and you're always using the same volume. And so I use a well-mixed sample. I put 4 drops on there, put it aside, let it dry. Then I'm going to stain one with the stain. I usually stain 2 with the Sudan black and I stain 2 with the Niger stain. And then I do an average. So this is kind of the Sudan black stain. I'm not going to go into detail about it, but it's a simple stain. You got 2 solutions. One you put on for 10 minutes, the other one, and then you rinse it off, and the other one you put on for 10 seconds, and you rinse it off. Niger stain is a little bit more complicated. You have a solution A and a solution B. But solution A is made up of 2 solutions. So you got to take 2 parts of one and 1 part of the other to make solution 1, and then you take the other one, which is solution 2. And then that solution you made up is for 30 seconds. The other one is for a minute. So it's a simple procedure. And if you buy the stain, the instructions are there. So then I take it and I scan my slide. So I scan down the slide, and then I scan up the slide, and I scan down the slide. I usually do 5 passes. You can do 3 passes, 2 passes, however many you want to do. I realize that the plant I work at is a 1.2 billion gallon a day plant. So it's a huge plant. I have the luxury of having a laboratory One laboratory just does bacteria, one does viruses, one does parasites, one does molecular microbiology, and I have 12 staff that can do that work. And so what I realize is a lot of operators don't have that luxury. Sometimes the operator is the one that does the lab, they do the cleaning, they're the mayor of the city and everything else too. So, but so you can just do 3 passes, just always do 3 passes. I have a really nice microscope. What my microscope does is as I'm scanning down the slide, it has an imaging software. And what I can do is I just click on that cluster and it surrounds it for me. And it lets me know what the area of that cluster. Then I scan down. If I see another cluster, I click it. It gives the area and it puts it over in a table for me. And at the end of my 3 passes, I have all this area these in the table and it totals. So I get the total area of PAOs per that slide. So I have a fixed volume, I have a fixed area, and it tells me how many PAO clusters, what the area of the PAO clusters was in that slide. If you don't have a microscope to have that kind of software, then you can just buy an eyepiece grid. It's a reticule that you can just put on the eyepiece that has these lines. And then each time you get to a cluster, you just want to count how many squares that cluster covers. And in this case, it's 19 squares. And then that's your number. And then you get to the next cluster, it might cover 5 squares. Then that's your number. Go to the scan down, you see another cluster, you count those squares. And then at the end, you might end up with 63 squares. That's your number. We're looking for trends. We're not necessarily looking for how many actual PO cells that's there. And so then what I do is I take that total area, and the reason why I use, there's 20 drops in a milliliter. So I use 4 drops, and that's 1/5 of a milliliter. So I take that total area, I multiply it times 5, and that tells me that total area in a milliliter. Then I divide it by the concentration times 1,000, and that gives me the total PAOs per milligram of VSS. The thing about PAOs is that gives them advantage is when it's warmer temperature, they grow so much faster in the summertime. They don't grow well though if the pH is greater than 7.25. So if you ever have any problem with GLs, because they're going to come in the summer if you have a biological phosphorus removal system, if you can increase the pH from 7 to 8, you'll get a dramatic drop in those PAOs. And I'm going to show you an example. This was one of our plants that we were doing. The Chicago Water Reclamation District has 7 water reclamation plants. Our largest is 1.2 billion, and this is one of our smaller ones. I think it's like 50 million. But this particular plant was doing a test trying to do biological phosphorus removal. The blue line is the number of GAOs. So in the summertime, you can see it's spring, it's starting to turn summer. GAOs just took off in the summertime. Our PAOs dropped dramatically in the summertime. But what happened was that there was a industrial spill, and it was a bunch of ammonia that ended up getting dumped into this plant, and it shot our pH up to 9, and then instantly the GAOs dropped just from that higher pH. And once they got out of the way, then our PAOs could take off and take over. And then we didn't have to worry about them too much because now it was October, November, December, and GAOs don't like the colder temperatures. And this is another example of one different one of our plants. The red line is GAOs, and you'll notice in the summertime they basically kind of dominate. But then as you get closer to the winter and the fall, they start dying off, and then your PAOs start taking over. And this is just doing that simple PAO GAO. We do GAO counts the same way. So volatile fatty acids are critical. You have to have them in an anaerobic zone. The reason why you need to have enough carbon is because you have to have enough bacteria to break down those organic materials so they can produce the VFAs. That's why carbon is so important. And so we need to have at least 7 to 9 milligrams of VFAs to remove 1 milligram of phosphorus. Another thing, the dominant VFAs are acetate and propionate. And so a lot of times when people are trying to supplement the carbon, they'll try to use acetate. But I suggest if you're going to supplement, that you use propionate, because GAOs like acetate. And so, but they don't like propionate. So this will help you prevent getting those GAOs. Another thing too is PAOs are sensitive to disturbance. They like a consistent and a stable load. And so I don't care how hard you try to maintain the right conditions for biological phosphorus removal. There are certain things in your treatment system that are kind of out of your control that you're not going to be able to handle, and that's the influence that's coming in. We don't know from day to day sometimes what's going to come into our plant, and so you're always going to have to need a backup. There's an optimum COD or carbon loading. For COD to phosphorus ratio, you need about a 40-to-1. For BOD to phosphorus, about a 20-to-1. But understand this, more is not better. Just, you don't want to overdo it, because if you overdo it, you'll end up having tremendous problems with filamentous bacteria. And I'll show you an example. One of our plants just didn't have enough carbon, didn't have enough organic material coming in. So what they did is they bought an external source. So it's called Micro C. And so they started adding it. They had 2 trains, a test train and a control train. And so they put the Micro C on one train, and then they didn't put it on the other one. The red line is the one that didn't have the carbon. The blue line is the one that did have the carbon. So and these are PAOs that I'm counting. The PAOs just began to increase. As soon as they stopped adding the carbon, the PAOs died off. But guess what else increased? The blue line is the PAOs. The orange line are filamentous bacteria. So the filamentous bacteria count increased right with the GAOs, because they had all this extra carbon. So you don't want to have too much carbon. And this was the filament type 021N. It just dominated. And you're going to find this one's going to be very, very common in biological phosphorus removal systems, because you have that, they like organic acids. This particular filament, thioretropics, they like organic acids. And so you have a lot of organic acids when you have this anaerobic zone going into your aerobic zone. But what I found out is that this pHb to poly-P ratio is the best indicator of how well the system is working. Because there's like 75 different genera of bacteria that can store pHb, but they can't all take up phosphorus. So if I'm looking at my anaerobic zone and I see a whole lot of pHb, but then when I get to the aerobic zone, it's only a little bit of polyphosphate, then what that tells me is that I've got a whole bunch of other things in there that's taking up pHb. Because this pHb to poly-P ratio for PAOs is about a 1-to-1 ratio. So if I got a whole lot of pHb and only a little bit of poly-P, then that means I either got a lot of GAOs in there too, and PAOs, GAOs, or some other microorganisms that like to store pHb. And so what that tells me is that that environment is not conducive to only PAOs in my anaerobic zone. So here's our Stickney plant. This is our 1.2 billion gallon a day plant. We retrofitted it for BioP. It's huge. We drive from primary to secondary. We had 4 aeration basins. You can see that with the lines, those are the aeration basins. Each aeration basin has 24 clarifiers. So we retrofit this plant for BioP. So you can see the sort of the dark areas is where the air was turned down. There's no baffles, so the oxygen doesn't know that it's supposed to stay on its side of the line. So you've got this kind of fuzzy zone in the middle there where it's kind of low DO, and that's why we were having some filamentous bacterial problems. And so if you look at this image, the return sludge line, they turned the air off. They turned it off in the mixing channel, and then in the feed channel. In each one of our aeration basins has 8 tanks. And so each one of these separate tanks, the air was turned off at that first half of the first pass. And so we collected our sample here at this red dot from the anaerobic zone, and then we collected our aerobic zone sample from the end here. And then we monitored the PAOs and we compared it with the outfall total phosphorus. And so as our polyphosphate, as our PAO number went up, you can see our outfall phosphorus went down. Once our PAOs dropped, as it was getting towards spring and summer, and it was probably because PAOs were coming, then our phosphorus was, our effluent phosphorus increased. So biological phosphorus removal is not going away. It requires a little bit more skill, a little bit more than just kind of letting the thing do its own thing. And we're kind of in a conundrum here because we've got to remove phosphorus, we got to remove nutrients, but the bugs need nutrients. Your biological phosphorus removal has one set of conditions that it needs. Your nitrification has another set of conditions that it needs, and just regular activated sludge has a set of conditions that it needs. And so it's going to take a little bit more skill. And so just looking at these things under mind, even if you don't do counts, just you can kind of get a sense of the abundance of them. Or if nothing else, just look for GAOs. And so just look and see if my GAOs are increasing. If your GAOs are starting to increase, then you might want to kind of raise up your pH, and that'll help prevent them from taking over in your system. And so with that, I think I'm done. I tried to talk really fast, stay on time, and if you have any questions, I'll be glad to answer. Was that supposed to stop at 4:30?
Thank you, Tony. Sorry for that mix-up again.
Oh no, I'm so sorry. I thought I'm just picking through the slides, thinking everybody was looking at my bugs. Okay.
I am just so impressed by your knowledge and that how much enthusiasm you have to explain it and keep it all straight. I love it. I love the analogies too on the PowerPoint slides. Yeah, it was fun. I'm looking and I'm not seeing any questions.
Do you do a lot of BioP there?
In some areas we do. You know what? My computer doesn't have the questions, Tony. Mm-hmm. So Stephanie's going to ask you the questions. And anyone who wants to answer, if they have a BioP plant, that would be awesome in the chat.
Hello, and it's Stephanie here from EOCP. So I've got access to the questions, so I'll just read them out.
Okay.
Okay. Thank you very much for that presentation. Like Anna said, it was great. So the first question is, Brian, do you produce VFAs on site through sludge fermentation, or is it only through supplemental addition of carbon source such as Micro C?
No, yes, you can do it on site. You can do it in your, in one of our plants, we're using part of our primary to try to help to produce some VFAs. We also partner with, I think a brewery or some company that wants to give up their waste for free, and we'll take it for free. Yeah, you can, in some of the plants, we're actually fermenting our own, trying to produce our own VFAs. It's kind of hard because you got to have enough capacity to produce enough. And yes, we do have tours of Stickney, so it's a pretty big plant.
That's an incredible big plant. We were just commenting of that. 1.2 billion.
Yeah.
I have another question was that your presentation was amazing, and they wondered if the presentation was available for sharing.
I'm sure if I can send it. I don't know how you want me to give it to you, but I can send it to you or to Stephanie or whoever. I can send it to you, and you can make it available to them.
That's
or
so
they can
nice.
Or they can email me for that email address, and I can send it to them.
Thanks so much, Tony. Stephanie will be in touch with you to share that with the delegates. Mm-hmm. That's great. This is another question. What is the main control of this process step, feed aeration or pH control?
The main control is making sure you have a fixed anaerobic zone. You got to have a true anaerobic zone, a true aerobic zone. So the main thing is to make sure you have enough organics and make sure you have a true anaerobic zone. I had a client call me one time because his BioP system wasn't working. And so what had happened is the consultant built the plant, made a slide, saw a bunch of blue dots on it, told him he had PAOs, and went on about his way. And so when I came there, I looked at samples of the anaerobic zone, samples of the aerobic zone, and they looked exactly alike. I saw a bunch of happy aerobic protozoans swimming around and having a good time. And they're not supposed to be in this anaerobic zone. And then I asked the guy, I said, are you monitoring this zone to make sure that it's anaerobic? He said, no, it says anaerobic. I said, it's not going to be anaerobic because it says anaerobic. You have to make sure that it's anaerobic. Just because you call it an anaerobic tank, it's not going to make it anaerobic. But what had happened was that his anaerobic, he had anaerobic aerobic grit chamber prior to the anaerobic zone. So he's got this aerated water going into this anaerobic zone, so he wasn't able to maintain anaerobic conditions. So the most important thing is that you have to have anaerobic conditions, followed by either an anoxic or aerobic condition, and then aerobic conditions. You got to make sure you have enough carbon to produce the VFAs that's needed. And so step feed is not, is more of a aeration basin control, but it's not going to do that much as far as the PAOs.
Excellent. I just have one technical issue I'm going to sort out, but there's a few more questions, Tony, if you can bear with me. Chris, it's not updating on the computer, but on the screen down below I can see more questions. No, I can do all those. I just see more on the screen below than my computer screen. I just was going to start with Mark Carey. Hi, Mark. And then Brian. I've got yours in the queue.
Hello. There we go. I'm on. Okay, so we've got another question. Do you produce VFAs on site through sludge fermentation, or is it only through supplemental addition of carbon source such as micro C? I think we've just answered that one, haven't we?
Yeah.
Okay, so the next one is, do you use indicators such as ORTH-P release in the anaerobic zones as performance indicator of the EBPR process?
Yes. So you have to get good phosphorus release. But you can get good phosphorus release and still have issues with GAOs, because your PAOs in your anaerobic zone are going to release phosphorus. And so it will help to at least look to see if GAOs are present. But yeah, that's a very good indicator. We should monitor phosphorus release in the anaerobic zone.
Okay, thank you. And Mark says, I saw acetate mentioned on one of the slides as a VFA or PAOs, but is there a preferred or optimal form or type of VFA for these bugs which will optimize their ability to take in phosphate?
Yeah, the dominant VFAs that PAOs use is acetate and propionate. So if you're gonna supplement it, I would recommend propionate, because acetate, the GAOs like it too, but they don't particularly like propionate, but your phosphorus bugs do. So the form of it would be propionate or acetate.
Okay, thank you very much. And I believe that's all the questions for today. Thank you very much.
Okay, you're welcome.
Yeah, I really enjoyed your presentation myself, and I'm sure everyone else has in the stream as well. Thank you very much.
Yeah, and I get to dig at my in-laws this way.
It was so enjoyable, Tony. Thank you so much. And just a lot of comments in the presentation that everyone really enjoyed it. And your style of presentation, it helps us remember it and laugh as we go.
Okay. So just be in contact with me and let me know, and then I can send you the presentation and you can give it out to people.
Absolutely.
Okay.
Absolutely.
Okay.
Thanks so
All
much,
right.
Tony.
You're welcome. Bye-bye.
Have a great day.
Okay.